I don't have a comment. Just let you know how to
We're not fighting because I guess it's one way. Like I'm getting mad at him.
it's just like, I don't understand.
Okay, we can get started now. When we finished, last time we finished talking about the purines, now we're going to begin with the pyrimidine biosynthetic pathway. And the first thing that we're going to do is just go through this pathway, and then after we've gone through the pathway, we're going to look at a couple of the enzymes. We're not going to look at many enzymes in the pathway as we did for purines. We're just going to look at these two enzymes at the beginning of the pathway because they're both interesting. This one is mechanistically quite remarkable, and this one is a very important regulatory enzyme that has been studied a great deal. So the first thing that we notice with the pyrimidine pathway is that it doesn't start with a big platform to which the pyrimidines are attached. Remember that with purines, we started with the PRPP platform. And then the purine base was built up step by step on the platform. And we went through a number of steps, 10 or 11 depending on how you count. In this case, however, it's just the opposite. The pathway starts small and builds up the purine base, and only then does it get attached to the PRPP. So it's the same PRPP, but the pyrimidine base is generated first and then attached to it. So we'll just go through this briefly. So here's the beginning, ATP and glutamine. So right away you know that you look at this and you say, okay, the glutamine is there because it's going to be donating the nitrogen, the amino group. And indeed that's what you see here. The amino group is attached to a bicarbonate, the bicarbonate up here. So the amino group of glutamine is attached to bicarbonate using the energy of ATP hydrolysis. And we'll go through this mechanism and you'll see how that happens. So ATP is used here to energize the amidation of the bicarbonate. Then the second ATP is used, as we'll see in the mechanism, in order to activate this substrate, forming the carbamyl phosphate. And the carbamyl phosphate then enters the next step, where this phosphate that was used to energize the substrate drives the addition of aspartate. And so here the carbamate has been added to an aspartate. And you can already see that the ring is almost complete already. So another unusual thing is that multiple atoms are added at once to build up the pyrimidine ring very, very quickly, rather than mostly one atom at a time, which is what happens with the purine. So then, these two steps basically finish off the ring after the atoms have been added. In this case, the elimination of water here closes the ring. And then here, this single bond is converted into a double bond. and that's the dihydroaurate dehydrogenase reaction. So this is oxidized to a double bond. And then here's where the PRPP comes in. The pyrimidine ring has now been formed. The PRPP comes in and the pyrimidine ring is attached to it, and that's what you see here. After the attachment, this is arotate, remember. After the attachment, then uridylate is produced by removing this carboxyl as CO2. And so here you have uridine, and then the kinases that we've talked about before, the uridylate kinase adds one phosphate, and then the third phosphate is added by nucleoside diphosphate kinase. So those two kinases phosphorylate this up to the UTP level. And then this needs to be converted to cytosine, cytidine 5' triphosphate, CTP. So UTP to CTP takes this carbonyl and converts it into an amino group. And you can look at this and you immediately say, I know what that reaction is. It's an aminotransferase reaction. Here we have a carbonyl. In this case, it's not an amino group because there's been a tautomerization, but it's exactly this type of reaction. And so you ought to be able to go through and write out the steps of the reaction for this particular case. There are a few differences because the substrates, of course, are different. There's, as I said, tautomerization going on, and some of the geometrical issues are a little different. But for all intents and purposes, it's an identical type of reaction. So that's just an overview of the pathway. All of the reactions really are familiar. This is dehydration, this is oxidation, and so forth, the decarboxylation, and as I said, we know this one. So we won't talk about these reactions anymore, but let's go now and talk about the carbanyl phosphate synthetase reaction, because that's really quite a remarkable enzyme. In fact, this enzyme, carbanyl phosphate synthetase, is probably the most awesome of all of the channeling enzymes. This channel that you can see, there are 3 active sites in this enzyme. This channel that goes all the way down the enzyme from this site, visiting this site, and then coming down here to site C, is 96 angstroms long. That's the length of 28 base pairs of DNA, just to sort of get a comparison, to get it in context. A very long channel, and we're going to just go through the reaction mechanism in order to see how that works. So the first thing, once again, a familiar reaction. Glutamine donates its nitrogen as an ammonia. And as you can see here, the ammonia is produced at site A and obviously has to travel to site B. So here's site A where the ammonia is produced. It has to travel down here to site B. And site B is where the carboxyphosphate is produced. This being the carboxyphosphate here, carboxyphosphate. So this step, the purpose of this step obviously is to activate the bicarbonate in this form so that now ammonia can attack this carbon, this being a good leaving group, and end up with our carbamate. The carbamate then is produced here. The carboxyphosphate is produced at this site. The ammonia comes into the bound carboxyphosphate, attacks it to produce this carbamate. And then the carbamate travels down the tunnel to site C, where there is a bound ATP waiting for it. And the carbamate is then activated to form carbamyl phosphate. So this single enzyme does multiple transformations. You can see that there are two activation steps. First, the bicarbonate is activated by ATP, and then the carbamate, which was formed using the energy of this ATP, this is then activated by ATP to produce the carbamyl phosphate, which is the substrate for aspartate transcarbamylase, in which the carbamyl phosphate is added, the carbamyl phosphate here is added to the aspartate. So let's look at the aspartate transcarbamylase enzyme. We're not going to look at the mechanism now. This time we're going to focus on the regulation of the aspartate transcarbamylase. We're looking at this enzyme because it is allosterically regulated and is one of the best studied enzymes in the entire pathway from the standpoint of regulation. And this regulation should be familiar to you because all of this looks very much like hemoglobin, right? And you've all studied hemoglobin and the sigmoidal behavior, the sigmoidal binding of oxygen as a function of oxygen pressure. You've all seen that. You know what cooperativity is in the context of hemoglobin. So we're going to look at these same principles, only here they're in terms of regulation of an enzyme. First of all, what we're going to be talking about here summarizes experiments that were done in the lab, in a test tube, with purified enzyme, which was presented with various activators, various regulatory molecules, and with substrate and so forth, in order to characterize what the nature of the regulation is for the enzyme. So don't try to fit this to an in vivo, to a situation in the body, because of course you don't have variations in substrate concentration and so forth like this in the body. This is just an experimental circumstance, but it tells us what the nature of the regulation is for this enzyme. So you remember from hemoglobin the T state and the R state. The T state being the state that binds substrate poorly, binds oxygen poorly. The T state here binds substrate poorly, so aspartate is the substrate. Remember what we're looking at. We're looking at the enzyme that catalyzes the addition of carbamyl phosphate to aspartate. So aspartate is one of the substrates of this enzyme. And so this is the aspartate concentrations that were used in this experiment. Concentrations from close to 0 all the way up here to 30 millimolar or more of aspartate concentration. And in the absence of any regulatory molecules, just the enzyme all by itself, but of course in the presence of carbamyl phosphate, to look at the production of the carbamyl aspartate product. low concentrations of aspartate, and in the absence of any regulatory molecules, this enzyme is in its T state. In the T state, it binds its substrate poorly, and because it has a high Km as a result of binding poorly, the Km is out here at a very high value, and so this reaction, if you extrapolate from the very beginning here, would be a very unresponsive reaction, a reaction that does not go quickly until you have very, very high concentrations of substrate. But of course, this is a sigmoidal. This is a cooperative enzyme. And so, as the aspartate concentration is raised, and as some aspartate binds to the enzyme changes conformation so that it can now bind aspartate better, so that the next when the next reactant comes in, the carbamyl phosphate, the reaction can go better. And so the higher the aspartate concentration, the better the reaction goes. It's of course a multi-subunit enzyme like hemoglobin, and so you get this cooperative effect when you bind one aspartate and then another to the enzyme. And so you can see the sigmoidal curve that develops as a result of increasing aspartate concentration as a result of the cooperativity of this enzyme mechanism. So this is the unregulated curve. The more aspartate that binds, the more active the enzyme becomes. However, this enzyme is one of the first enzymes in the pathway leading to the pyrimidines, U and C. And so it's subject to feedback inhibition. So here is C, feeds all the way back here to inhibit this reaction. ATP, on the other hand, accelerates this reaction. So these are opposing effects. This makes sense. It's the final product of the pathway, and so it inhibits this if its concentration builds up. ATP, on the other hand, is at the very beginning of the pathway, and if there's plenty of ATP around, then it serves the cell well to produce more CTP and UTP, more pyrimidines in other words, because apparently there's a high concentration of purines around. So high concentration of ATP accelerates this reaction. The high concentration of CTP inhibits the reaction. And so you see here, if you put CTP in there, it's still sigmoidal, but now it's at a much slower rate. The Km is increased substantially because CTP binds preferentially to the T state. So again, remembering your hemoglobin, aspartate binds best to the R state of the enzyme. It has a higher affinity for the R state of the enzyme. And because it has a higher affinity for the R state of the enzyme, the binding of aspartate gradually converts the enzyme into the more active R state form. But CTP binds better to the T state of the enzyme. So if there's CTP around binding to an allosteric site, it now stabilizes the enzyme back in the T state in spite of the fact that there's a lot of substrate here. And because the enzyme is stabilized in the T state by the binding of CTP, the reaction is slowed down. So that's the feedback inhibition of CTP. However, if the ATP concentration is high, it counteracts the effect of CTP because the ATP binding in its regulatory allosteric site, ATP binds better in its site to the R form. So it stabilizes the enzyme in the active, or the more active, R form. And so these are the various influences that go on here. Now, of course, in the cell, you'll never have a situation where you have only CTP and no ATP. And so because this is a laboratory experiment, you see the extremes here, but what is happening in the cell is you always have some CTP and you always have some ATP, and their concentrations will vary. And so there's going to be a curve similar to this that gets a little bit more or a little bit less. So remember, whenever we talk about regulation, we often talk about on and off and that sort of thing, but in the cell, everything is controlled so finely that the variations are actually very small. As soon as ATP concentration begins to rise, the reaction accelerates, producing more CTP, and the moment CTP concentration begins to rise, the reaction slows down. And so it's held in a very narrow range in vivo. All right, so that takes us to the end of the purine synthetic pathway and the pyrimidine synthetic pathway. And those pathways, as I said before, are the legacy of the RNA world because they were attached to PRPP, which is ribose. Now we're going to look at the add-ons resulting from the ascendance of DNA as the genetically important molecule in the cell. And so rather than invent a whole new pathway for synthesizing DNA, obviously from an evolutionary point of view, it was much easier to take the ribonucleotides that were already being produced by metabolic pathways and convert the ribonucleotides into deoxyribonucleotides by a ribonucleotide reduction reaction. So this is added on to the reactions of the RNA world after the synthesis of purines and pyrimidines that already evolved. And as I mentioned before, there are going to be two things that have to be done. Number one, you have to reduce the ribose from ribose to deoxyribose, and the U has to be converted into T. So we're going to be looking at both of those reactions. So just to keep in mind that we are talking about a reduction reaction here, reducing the sugar. Here we have the nucleotide diphosphates being converted to deoxynucleotide diphosphates. And this is a reduction, the hydroxyl form to the deoxy form. And so there has to be, obviously, a compensating oxidation. So the reduced form of ribonucleotide reductase, the enzyme responsible, has to be converted to the oxidized form. Now you've gone through all this with the TAs in section already, so I'm not going to spend a lot of time over it, but I just want to review it quickly. As you can see here, this oxidation of the enzyme leaves it in a state that has to be reduced once again, and so forth all the way down, and ultimately you see that what is driving the reduction of the ribose here is the oxidation of NADPH. And so it's the reducing power of NADPH that is ultimately responsible through either this pathway or this pathway for the reduction of the riboses. So now we're going to look just briefly at the ribonucleotide reductase mechanism. It's a remarkable enzyme. Its detailed mechanism is still not understood completely, but the outlines of it are very clear. There are a lot of complexities involving ribonucleotide reductase that we won't talk about. We're looking here at one form of the enzyme, the tetrameric form, with 2 alpha subunits and 2 beta subunits, and we'll confine our discussion to the tetrameric form. But part of the regulation actually involves changing the state of the enzyme from monomer to dimer to tetramer and to hexamer, all of which have different activities. But we're not going to get into that. We'll look just at the tetrameric form, and we'll focus on the mechanism, and then we'll just look at the regulation from a conceptual point of view, not the structural point of view that involves changing the subunits. So the reduction is driven by a tyrosine here that's in a radical form, a radicalized tyrosine, and it's maintained in its radical form by the iron in the beta subunit as shown here. This tyrosine over here in the three-dimensional model is quite far away from the active site, which is up here. And so there has to be a way in which electrons are conducted from here to here and back. So when we look at the mechanism later, keep in mind that we're dealing with actually great distances here. This diagram just shows you the structure of this enzyme in a ribbon diagram. Here are the 2 alpha subunits that you see here, and the 2 beta subunits do not just contact the alpha subunit like that. This is very diagrammatic, but instead the 2 beta subunits are like this, and they fit into the 2 alpha subunits. So we have one beta subunit in front of the other here. You can see the 2 different colors. Okay, so just going through the mechanism briefly. This X, which is probably a cysteine at the active site, probably this cysteine 439 here. So that radicalized X corresponds to the cysteine sitting up here that was generated ultimately by this beta subunit tyrosine radical. And so the tyrosine radical here is allowing electron pressure to radicalize this cysteine 439. So we're not talking about this tyrosine radical when we look at the mechanism. We're looking at a cysteine radical up here. And that radical, they're not entirely sure, that's why it's called X here. That radical abstracts a hydrogen atom. It abstracts a hydrogen atom, not a proton, not a hydride ion. A hydrogen atom, which is of course a free radical too. And so the hydrogen atom plus this free radical gives you a neutral molecule as shown here. And now instead you have a radical up here. So this is the last step in the propagation of the free radical all the way from the tyrosine down in the beta subunit up now to the substrate. So we have the radicalized substrate here. And at this point, this gets protonated. So a proton comes from here and protonates this OH to produce this. And once again, this is a proton transfer, not a hydride ion transfer. One is used to seeing hydride ion transfers for enzymes that have active site sulfhydryls. So this enzyme mechanism is unusual all the way along. This is a proton transfer, and so we just have a positive charge here, but no oxidation reduction has taken place. Okay, the purpose of this reaction is to reduce the riboses, but it's not at this step. This is just a proton transfer in order to charge, get a charge here, which makes it easier for the water molecule to leave, leaving this charge behind. And it's at this point that a hydride ion reduction takes place. Here a hydride, a negatively charged hydride ion, comes in to this positively charged carbon, adding a hydrogen to it. Reducing it as shown over here. The purpose of the water leaving at this point is, of course, to make the carbon here a good target for this nucleophilic attack. And this carbon is stabilized with a positive charge by a resonance structure involving this hydroxyl over here. So here you can see the resonance of this radical and this positive charge migrating between these 2 points. So these are equivalent atomic structures with the electrons moving around in 2 different resonance forms, and that stabilizes this, allowing this attack to be efficient. So after this hydride ion comes over here and neutralizes this positive charge with a reduced carbon now, gained an electron. That's why it's gone from a plus charge to a neutral. At that point, this radical takes back the hydrogen that it originally lost here at the beginning of the mechanism to restore the ribose ring as deoxyribose now. And then the enzyme itself, which has become oxidized during this cycle, the ring has become reduced obviously, but the enzyme has become oxidized and has to be reduced for the next cycle by the rationale shown here on this slide. So that's the mechanism of the reaction. Let's look briefly at the regulation of this reaction. Remember, as I said before, a nucleotide imbalance. The cell is set up to accommodate a particular balance of concentrations of nucleotides. They're not all the same concentration, but the balance is important. And if the cell experiences concentrations that move away from the established set point in the cell, it becomes mutagenic. Because with an excess of one particular type of nucleotide, it becomes more likely that that nucleotide could be inserted erroneously during DNA replication. So a balance has to be maintained, and we saw previously how symmetry is important. ATP driving guanine production, guanylate production, GTP driving adenylate production, symmetry, to keep the purines in balance. And here was a broader view of the regulation of purine metabolism. Again, drawn so as to emphasize the symmetry of the regulation to keep the purines in balance. And we'll see that kind of symmetry coming up again, but with in a new context and with some new elements to it. So here you see two different types of regulatory site in the ribonucleotide reductase. There are the primary regulatory sites, so-called, and the substrate specificity sites. These are called the primary regulatory sites because regulation just makes everything faster or everything slower for all the nucleotides. So over on this side, ATP accelerates all of the reactions that produce CDP, UDP, GDP, and ADP. They're all accelerated by ATP. And once ATP has been reduced to DATP, and the DATP concentrations rise, then it slows everything down. It begins to inhibit all of the reactions carried out by this enzyme. So these are the primary regulatory sites which determine the overall activity of the enzyme. But in addition, the enzyme has the capability of being regulated to vary the rates at which individual nucleotides are produced so as to prevent imbalances from developing. So, in the presence of ATP, which accelerates the whole reaction, either ATP or DATP, as it turns out, can accelerate the specific production of dCTP and dTTP. So this is accelerated when there's lots of purine, but perhaps not quite enough of the pyrimidines. So the reaction, the enzyme as a whole is activated, but the pyrimidine producing reactions are activated a little extra. Notice that when there's a high concentration of DATP, DATP in this case, as opposed to ATP, the reaction is actually, the enzyme is actually slowed down. So ATP accelerates everything as a whole. All the reactions of the enzyme are accelerated in the presence of ATP. And the presence of DATP, if there's an excess of purine, accelerates the pyrimidine reactions a little bit extra, as I said, but inhibits the overall activity of the enzyme. Why does it inhibit the overall activity of the enzyme? Because we have a lot of it. And if we have a lot of it, it means it's not being used as fast as it's being produced, and so we want to slow down the production of everything. And so when there's a lot of this, the production of everything is slowed down, but because this pathway is activated because of this imbalance, everything is slowed down, but what little synthesis takes place is going to be primarily pyrimidine synthesis to try and restore the balance. So the excess of DATP tells us two things: that there's too much nucleotide overall, and therefore we need to slow everything down, but also that there isn't enough pyrimidine to balance the amount of purine. And so we slow everything down, but what little synthesis is happening is largely the pyrimidines. So the pyrimidines are produced, DTTP is produced, its concentration builds up, and so it feeds back and inhibits its pathway while accelerating the purine pathway here of GTP. As GTP concentration builds, DGTP concentration builds up, it feeds back to inhibit its own pathway while accelerating the production of DATP from ADP. And of course, when DATP concentration builds up, then what I talked about at the beginning takes place. The overall activity of the enzyme is reduced and the DCTP, the production of the deoxypyrimidine nucleotides is accelerated. So this pathway, as you can see, produces the pyrimidines. Yeah.
The DATP initially activates the production of both pyrimidines, correct?
Yeah.
But it's not the DCTP that regulates the production. Oh, I see. So then, and then the DTTP will activate the production of DGTP. But why would DGTP activate the production of DATP?
To balance it, yes. Because this is the only reaction that's being activated by the DTTP. And so as soon as this starts being produced,
But wasn't there an initially high concentration of DATP, so wouldn't that just end up like, sort of like a,
yes, but
making lots of ATP in the, like DATP, high DATP makes lots of DATP. That's like kind of what I'm
yeah,
worried about.
no, you're absolutely right. So there's a lot of this, so the enzyme is slowed down, and this is accelerated. But if this regulation is successful, you're soon going to have too much of this. You don't want the pyrimidine or the GTP concentration to rise out of balance with the DATP. And so this is designed so that as these concentrations build up, they eventually become high, and so it comes back and has to keep the DATP concentration high. If this concentration is high enough to activate, that means that now you have successfully synthesized a lot of this, and you don't want it to go too far without bringing this along.
But it still seems like, well, okay, so you'll make more DATP.
So as you make more DATP, you shut the enzyme down.
Okay,
More
so
and
then
more
that's
and more.
just from any like additional. I see. Okay, I got it.
So it's a loop. It's a loop. Everything is activating and inhibiting directly or indirectly everything else. And so with everything getting in the act like this, that makes sure that everything stays in balance. And when you look at this, it's not going to be perfect. The symmetry, for example, in this diagram is not perfect. It's not absolutely perfect. And if you look here, this is for E. coli. In mammals, it's a little bit different, and it's not a perfect symmetry, and that's because of the fact that this resides in the cell where there are other reactions with regulation involving these kind of molecules going on. And so it has to be in the context of overall cellular metabolism. And in fact, we'll look at that right now here, because you notice that here there is no individual regulation of DCTP and DTTP to keep them in balance. But that's okay, because there's this separate set of reactions down here which balances the DCTP and the DUTP. And the same thing here with the GDP and the ADP, DGDP and DADP. Their balancing is affected by the prior balancing of the ribonucleotide versions. So you remember that this symmetry of regulation involves the ribonucleotides, and that obviously, you're starting with something that's balanced. and that helps this to stay in balance. But these are not regulated, the balance of these is not regulated in the ribonucleotide reductase. They're regulated by the deoxycytidine deaminase reaction, and you can see it makes a lot of sense. If you have a lot of CTP, DCTP, it accelerates the production of DUTP in order to make sure that DUTP is in balance with the DCTP. But if the DUTP gives rise to an overabundance of DTTP, then it comes back and inhibits this same reaction. And so this acceleration and this inhibitory activity, when they're in balance, ensure that the balance of DCTP and DTTP is maintained. Let me just draw one item here to your attention. This DUTPase. DUTP. Bad. Why is DUTP bad?
Mismatch, like in the UG case pair instead of.
Won't be a mismatch. Won't be a mismatch.
Wait, not mismatch. Sorry. When deamination. Yeah. Sorry. Cytosine gets deaminated to.
Okay, but you're on the right track, but we're not talking about deamination of C in DNA, which is what we talked about before. This is part of a metabolic pathway.
Didn't it lead to like different base pairing when C was deaminated to U?
Yes, that's because the deamination occurred when it was in DNA. And the C is paired with G, and then it gets deaminated and says, what's going on? This is a metabolic pathway. DNA is not involved yet. So the CTP is not paired with anything, and so the deamination here just produces DUTP. But it's not disrupting any pairing because no pairing has happened yet. It's just floating around as a free nucleotide. Yeah.
Can it get incorporated into DNA?
Exactly. DUTP, if it's at a high concentration that's produced by this pathway, would get incorporated in place of DTTP. This is what we want to get incorporated into DNA, DTTP, not DUTP. And so you can see there's a bit of a problem here. The RNA world is switching over now to the DNA world, and so the ribonucleotides have to be converted into deoxyribonucleotides. And of course the ribonucleotide, in this case, for this particular nucleotide, is UTP. So UTP has to be converted to DTTP. And so we start here with UDP, and that's converted by ribonucleotide reductase, as we saw, to DUDP, and then it's kinased up to DUTP. And then it must be converted from T into U, and I think that's the next thing we're going to talk about. Yeah, that's the next thing we're going to be talking about, is the conversion of the T of the DU to a DT. That's the famidylate synthase reaction here, the conversion of U to T. But because this whole reaction started with the RNA world, it passes through the DUTP before it can become DT. Yeah.
How can the cell make a mistake of incorporating U in the place of T?
Well, the polymerase has to recognize the difference between a U. The only difference between this and T is a methyl group.
Yeah, so wouldn't that cause a lot of fraying and it still edits it out anyway?
A little bit, yeah. But why would it cause fraying?
Because of the active site, the methyl group is not only UMP, so it still doesn't fit right.
Almost. UTP differs, DUTP differs from DTTP by a methyl group. The Watson-Crick hydrogen bonding is just fine. But because of the lack of the methyl group, it wiggles a little bit, which will make it more mobile, and therefore you're right, it's going to fray a little bit more and will be corrected to some extent because of that. But in addition, the polymerase has to discriminate. The UTP, remember, can fit in the site perfectly well because it's smaller, it's not bigger. So it fits in perfectly well, so it's a really difficult job. If there was a high concentration of DUTP in the cell,
Okay, it's high probability.
then you would get an intolerably large number, that is to say, 1 in a million or something, of incorporations of UTP instead of DUTP instead of the DTTP. And so the enzyme does a marvelous job, and repair does a marvelous job of preventing this, but it would still go up. And so the important thing here is that there is a DUTPase that's very active. And the moment the DUTP is produced, the phosphates are removed, converting it into DUMP. So this seems illogical that you've got it phosphorylated up to a triphosphate, and the moment it's produced, you take off 2 of those phosphates to produce DUMP, you convert that to thymine, and then you have to phosphorylate it up again. And so that is the holdover from the RNA world. Converting RNA world to DNA world involves some uncomfortable reactions like this. And that's what we'll pick up next time.